Okay, happy Wednesday. So we're about to start metabolism today. So thinking back, you've already learned a lot this semester. We've talked about what microbes are and how we classify them and how we see them through a microscope and their structures. Last time we talked about how they grow. And maybe that's a little bit out of order because, of course, microbes can't grow unless they have energy, right? So that's our topic for probably the rest of this week. And then we'll get into thinking about, well, what's that flow of energy or flow of information? How do they know what structures to make? That's genetics, and we'll get into that next time. So maybe we do growth before, maybe in a mixed up place, but anyway. So metabolism, how cells acquire energy. We are not going to jump right into pathways. I'm going to spend a little time reviewing kind of the things that you need to understand the pathways. These should be things that you've had before, but it tends to be those kind of abstract things that maybe you've forgotten. So we're going to review a little bit things like oxidation reduction reactions, electron carriers, little terminology around enzymes, things like that. Before we get into our catabolic pathways, we're gonna take a really kind of big picture approach to this. So a much different approach than your biochem professors, where you're gonna be memorizing structures and enzymes. There is definitely an elegance that you learn to appreciate when you look at those structures, but we're not going to take the time to do that. So we're going to have kind of an overview that we're going to take, like when would an organism use this pathway? When would it use another pathway? So we're going to kind of differentiate between the pathways, hopefully respiratory pathways. We're going to pay particular attention to those that are uniquely microbial. Okay, so things that bacteria do that you may not have talked about in other classes, so fermentation in particular, and then end with a discussion of what if it's not glucose that, what if it's not glucose that the organism is using? What if it's some other macromolecule like protein or lipids? All right, so that's where we're going. Let's start again, a little bit of review. Those two terms, anabolism and catabolism. We don't want to get those mixed up. An anabolic reaction, that refers to a reaction that builds molecules. Okay, or biosynthetic reaction. Okay, so any reaction that builds molecules is going to require energy, so we call those endergonic reactions. A reaction that's catabolic is a reaction that breaks down molecules. Okay, so energy is going to be released from this kind of reaction, so we call it an exergonic reaction. And if you maybe tend to get those two terms mixed up, there's a little mnemonic that I came across, ABCD. So anabolic builds, catabolic degrades. I don't know, maybe help. But these reactions in the cell, and anabolic reactions and catabolic reactions have to be really tightly balanced. You can't have too much of one and not enough of the other. All right, and just so a little graphic of that, remember, we're always talking about bacterial cells unless I say, oh, but in eukaryotes, such and such happens. And sometimes I'll make a contrast, but always assume that I'm talking about a bacterial cell. So here's a bacterial cell. It's taking in nutrients. And remember, it's not going to phagocytize nutrients. It's got a cell wall, got to absorb them some other way. Maybe it's got to secrete enzymes to break them down first. You'll be talking about that in lab in a couple weeks here. But in any catabolic reaction is not going to be perfectly efficient. You're going to lose a little bit of energy, and that's typically lost as heat. Okay. Organisms have reactions that are more efficient than, say, a car engine, but they're still not perfect. So we lose some energy as heat. So gain ATP energy from catabolic reactions. There are precursor molecules maybe that are formed, and those can be used in anabolic reactions to build macromolecules. So maybe for storage, some macromolecule like a carbohydrate or maybe a lipid, a protein, nucleic acid. Those are the 4 macromolecules. Okay, cellular structures, energy storage, things like that. And again, maybe gonna not perfectly efficient. So these terms are always kind of, they trip me up a little bit, to be honest, but it's like, wow, you see them and you gotta kind of understand what they mean. So we have terms that describe how organisms acquire their carbon for metabolism, and also how organisms acquire their energy. And you see them, so I want you to be able to recognize them. So pay attention to the prefixes on all these terms. Troph just means kind of like to eat, basically. I think that's a similar root there. So if an organism acquires its carbon by an inorganic source, then it's termed an autotroph, auto meaning self. Okay, so the only really, there might be some other section, but inorganic carbon, that's carbon dioxide. Remember, if something is organic, it has carbon-hydrogen bonds. Okay, so CO2 has carbon, but not carbon-hydrogen bonds. So an autotroph gets its carbon just from the carbon dioxide in the air, and a heterotroph has organic sources of carbon. So in other words, I like to think of this as they eat stuff. Okay, and both methods are very common. Obviously, anything that photosynthesizes is going to be an autotroph. Plants, algae, cyanobacteria. Most other organisms are going to be heterotrophs. So, fungi absorb nutrients. We're heterotrophs. Okay. And then ways to describe the source of energy. Phototrophs, obviously photo, that makes you think sun. So they acquire energy again from the sunlight. And then chemotrophs are going to acquire energy from the breaking of bonds. The breaking of bonds where, those were the next 2 terms, organotrophs and lithotrophs, if it's from an organic source, and it's an organotroph. Sorry, the breaking of chemical bonds, that might help a little bit. And then lithotrophs, some inorganic source of those chemical bonds breaking. So the confusing, I think, confusing part gets to be when we mush these terms together for carbon acquisition and energy acquisition. So we talked actually already about how fungi are chemoheterotrophs. Okay, so they get their energy from the breaking of bonds and their carbon from basically absorbing nutrients usually. So again, we kind of squish those together. Most organisms, this is not common. Lithotrophs getting energy from an inorganic source. It's only some bacteria that can do that. So that's not terribly common. And they're not bacteria that are causing disease. So we're not going to worry too much about that. Oxidation reduction reactions, something that you've had probably lots of times before. Again, just sort of the abstract nature of it. It's confusing. I love this little YouTube video. It's a crash course chemistry YouTube video, and he talks about how these terms oxidation and reduction are kind of not the best terms. He talks about how we came at those, and he said, you know, well, we just got to learn this stuff because it's everywhere, and redox reactions are essential for metabolism. When a molecule loses an electron, then we say that it's oxidized. And when a molecule gains an electron, we say that it's reduced. So you could think about the net negative charge as being reduced, possibly. But I know it's weird when you gain something, it's weird that it's actually reduced. So a mnemonic that's probably most of you know or have heard of, oil rig. Okay, so oxidation is loss of electrons. It's all about the electrons. That's what he says in that video. I'll say that probably several times going forward here. So oxidation is loss of electrons and reduction is gain of electrons. Okay, so good mnemonic. I think the confusing thing happens is that I don't see where electrons are being transferred in this equation. So remember to follow the hydrogens. Okay, so protons often accompany electrons. Okay, so a hydrogen atom, for instance, you know that that's made of a proton and an electron. All right, so we do see the protons in equation. And whenever you see a molecule gaining a proton or gaining a hydrogen, remember that there's actually an electron in there too. So that's illustrated. Those redox reactions are illustrated, I think, as we talk about electron carriers. And by the way, that YouTube video, he talks like super fast in there. So I have to watch it a couple times, but it's like 7 or 8 minutes long. And it goes through some things that we don't really talk about, like oxidation states. But again, I think that is really helpful. It's something that you need to review. Electron carriers, we find 3 different electron carriers in cells. These are important because we can't just let electrons kind of go wherever they want in the cell because they cause damage. They have to be chaperoned from place to place. So you could think about nicotinamide adenine dinucleotide, NAD, as an electron chaperone, for instance. And NAD, this is in its oxidized state, NAD plus. It can actually carry a hydride ion. Okay, so let's write that here. It carries a hydride ion, which is actually 2 electrons and 1 proton. So when it's shuttling electrons, it's going to, in its reduced state, it's going to be NADH plus H plus. Okay, there's an extra proton left over. So it's actually carrying here a hydride ion. There's 2 electrons and a proton. Okay. FAD, flavin dinucleotide, flavin adenine dinucleotide, can actually carry 2 hydrogens. So it's in its reduced state, it's FADH2. And then NADP, nicotinamide adenine diphosphate. When we see that phosphate, that tags the electrons that it's carrying to help power biosynthetic reactions. So with NAD and FAD, those electrons are going to get shuttled to some kind of electron transport system and get turned into lots and lots of ATP. But with the NADP, those electrons, it actually can also carry a hydride ion. So in its reduced state, it's NADPH plus H plus also. Okay. All right, so we'll come back to that. There's a couple of pathways that have NADP, and so we'll call attention to that again later. Of course, the energy currency of the cell is ATP, a recyclable molecule. It recycles between carrying 2 phosphates and 3 phosphates. It has these high energy bonds between the phosphates, and when those phosphates are broken, that releases energy in a usable form. And then when again one phosphate, we call that when it being phosphorylated. Oh, sorry. When you add a phosphate, it's phosphorylated. Yeah, back to ATP. Okay, and that happens again. Depends how the cell acquires energy. It might be from the sunlight. It might be from the breaking of chemical bonds here. So there's 2 different ways that we describe that phosphorylation. You've probably heard these terms before. When I say how is phosphate added to ADP to make ATP, we're going to talk about several different ways here. The first is substrate level phosphorylation. Okay, so kind of don't get wrapped up in that actual glycolysis pathway here. What I want to draw your attention to is the way that we get ATP during this pathway, for example, during glycolysis. It's by substrate level phosphorylation. So that means the phosphate that's added to ADP to become ATP is usually cleaved off of one of the substrates in the pathway. That's not always true. It can be a phosphate in solution. But for instance, here in glycolysis, down here at the kind of later steps of glycolysis, this molecule 1,3-biphosphoglycerate, it's got 2 phosphates on it. Okay, so one is peeled off and added to ADP here to make ATP. That's our first substrate level phosphorylation. And then this phosphophenylpyruvate, here's another phosphate here that gets, again, added to ADP to make ATP. So 2 examples of substrate level phosphorylation there. So this happens during glycolysis as well as during the Krebs cycle. And we're going to look more closely at those pathways in a little bit. Another way that we get phosphorylations is by oxidative phosphorylation. And this is going to take place in electron transport chains, or your textbook refers to them as electron transport systems. So I'll try to use that terminology. And I think it's easy to get mixed up and say, oh, in oxidative phosphorylation, that's when oxygen is required. No, that's not precisely true, because a lot of bacteria are going to use something else other than oxygen as a final electron acceptor. They might use something like nitrate or sulfate. So it's not just oxygen. What's happening is that electron carrier NADH is getting oxidized back to NAD. Okay, and that's where that term oxidative phosphorylation comes from. These electrons get dropped off as some electron carriers in the electron transport chain, and that, as we will see and we'll look at more closely, is what powers the phosphorylation of ADP to ATP. Okay, so I want to plant this picture of a dam here. We'll come back to this later, but in the electron transport system, when those electrons are passed, the protons are kind of put outside of the cell. And a great analogy for that, those protons are potential energy. Just like this lake water here is potential energy. When it flows through that dam, those turbines are going to produce electricity. When those protons flow in during just a certain spot in the cell membrane, that powers the phosphorylation of ADP to ATP. So we will concentrate on that much more. The third way that phosphorylations take place is by photophosphorylation. That happens during photosynthesis, and we're not going to go into any more detail about that. I'll just mention it just out of, being complete. But again, no pathogenic organisms photosynthesize, so we're not going to worry about that. Okay, a little refresher on enzymes. Again, I hope that this is review. Enzymes, of course, are going to lower the activation energy of reactions within the cell. They're reactions that would take place anyway, but so slowly that life wouldn't be supported. So they make these reactions happen much more quickly. The enzymes are not going to be used up. They cycle back and go back and change another reaction. Enzymes work on substrates. Actually, here they call them reactants. So I'll put that in parentheses. Okay, we usually write the name of the enzyme on a little arrow of the reaction that it's catalyzing, and what we end up with are products. Okay, so sometimes in lab here in the next few weeks, we're going to be testing for a lot of different enzymes. And when I ask students what the substrate is, sometimes I get a lot of blank looks. So just remember that's what the enzyme acts on. How do you know if something is an enzyme? Well, if the molecule ends in ASE, like lyase or lactase, that's not really a good example. Anyway, if the enzyme ends in ASE, then you know it's an enzyme. Okay, there are enzymes that don't end in ASE, like trypsin, for example. But if something ends in ASE, then you know it's an enzyme. If something ends in OSE, then you know it's a sugar, glucose. Okay, so don't get those two mixed up. Let's see. So a catalyst is something that's not changed or used up in the reaction. So these are biological catalysts. Some enzymes are going to be just protein. Okay. But a lot of times enzymes need some other component that's non-protein for them to work. Okay, so if a non-protein component is necessary, then it's either called a cofactor, if it's something that's inorganic. Yeah, that's right there. And it's called a coenzyme if that's something is organic. Okay, so an example of a cofactor, some enzymes need something like, say, zinc or maybe iron or copper in order to, here's the example right here, in order to make that active site of the enzyme be the right shape. Of course, the active site is where the substrate binds. Likewise, if it needs something that's organic, it might be like a vitamin or something like that, in order for the active site to have the right shape, then we call that a coenzyme. Okay, so just a little terminology. When you talk about just the protein parts of the enzyme, it's correctly called an apoenzyme. And if you take the any cofactors or coenzymes, if you include those, then we call that the holoenzyme. All right, so thinking about enzyme activity. Oh, so this is just your a nice diagram, I guess, reviewing that substrates, and the gray part here is the enzyme. Substrates bind at the active site. Sometimes there's a little bit of induced fit, so sometimes the enzyme can change a little bit to fit that substrate just right. And then the enzyme is going to do something to change that substrate. Maybe it's cleaving it into two different parts. Okay, so here's a product that's different than what the substrate was. Okay, the enzyme isn't changed in any way. It can go back and catalyze the same reaction for a different substrate. And this is just another illustration of how, hey, maybe some cofactor or coenzyme is necessary so that the substrate will bind. That's very, very common. Enzyme activity can be dependent on environmental factors. So last lecture, we talked about how bacterial cells will grow maybe at different rates depending on the temperature or the pH. That really comes down to what's the ideal temperature or pH, for example, of the enzymes that are in the cell. If an enzyme is working, the fastest, it's like 37 degrees Celsius, then that organism is probably going to grow fastest at 37 degrees Celsius. So that's no surprise. So again, they have an ideal temperature. It doesn't necessarily have to be 37. It could be a different temperature. They have an ideal pH range, can be different for different enzymes, obviously. We have enzymes that work at very low pH in our bodies. Also, the substrate concentration, as well as the amount of enzyme molecules that are available, can make a huge difference as to how quickly the reaction goes. And that's will saturate to a certain point. So you have more and more substrate, the reaction goes faster, but then once you get to have the same number of substrate or more as the number of enzymes, it doesn't necessarily get any faster. So just recognizing there is a saturation point. And ideally, cells are going to be able to regulate a little bit when their enzymes are working or when they're not, just to have a little control over their metabolism. Commonly, we talk about competitive versus non-competitive inhibition. So with competitive inhibition, there's some molecule that's binding in the active sites of the enzyme that's keeping the substrate from binding there. Okay, so there's not going to be any products because the substrates can't bind to the active sites. That's competitive inhibition. A non-competitive inhibitor is going to bind at some allosteric sites. Allosteric basically means other than the active site. So it's going to bind maybe on the other side of the enzyme here, but that can still induce a conformational change in the active site so that the substrate can't bind. Okay. Also want to recognize that sometimes it's better to talk about it in terms of non-competitive control and not just inhibition, because here, so I'm getting a little feedback there. So here we've got the top one is allosteric inhibition, but sometimes there's going to be some allosteric molecule that actually changes the active site so that the substrate now will bind. And we could refer to it as an allosteric activator. So it can kind of work both ways. And lastly, thinking about, when enzymes are functioning and when they're not, there are feedback loops, which makes sense maybe in a biosynthetic pathway where you've got lots of different enzymes. And if the product is building up, maybe that's too much and the cell doesn't need it. So some of that end product will actually bind allosterically to the first enzyme and now make it so it doesn't keep making more and more product. Okay, so cells obviously don't want to spend more energy than what they have to, and if they've got too much of something, they have a way to shut it off. All right, so questions so far. That's kind of our background primer on metabolism. Keep in mind as we start looking at some pathways, we're going to pay close attention to how much ATP we start with and how much we get out of it. We're also going to look a little bit at some key juncture points. So again, we're not memorizing pathways, but we are going to take note of, okay, now the cell can either go this way or it can either go this way. It depends on this or that. We're going to look at what's actually produced. And when a cell might use one pathway and when it might use another pathway. It helps to follow the carbon. So if you're starting with a 6-carbon glucose, yes, it makes sense that we end up with 2 3-carbon pyruvic acid. And if we do lose some carbon, we're going to take note of where we lose that. Lastly, keeping track of those electrons that are being carried by our carriers. It's all about the electrons because that's what gets converted into the lion's share of the ATP during respiration pathways anyway. So we gotta follow those electrons. I like this overview summary of metabolism that was in our old textbook. So I'm gonna be showing this several times. Summary of glucose catabolism. Why do we always start with glucose? Because that's usually a cell's first choice when it's the most readily available energy source. So if glucose is available, it's almost always going to use that before it's going to use something like proteins or whatever else. And it's a simple sugar. So sugars that are more complex are often have to be chopped up before it can, funnel into some part of glycolysis. So glycolysis, you can see, is part of both respiration pathways and fermentative pathways. Okay, so we always start with some glycolytic pathway where sugar is split, the lysis of sugar. We'll talk more about fermentation. Usually, if a cell has some kind of electron acceptor here at the end, it's going to do some kind of respiratory pathway because it's always going to get more energy that way. With fermentation, there's an advantage, and we'll talk about why a cell would do it, but it's not really getting very much ATP from a fermentative pathway like after tacked on to glycolysis. So whatever it can, it's going to do either anaerobic or aerobic respiration. So that's going to go through some kind of Krebs cycle, lots of electrons coming off the Krebs cycle, a few from glycolysis as well. Those electrons get transferred to electron transport system within the cell's membrane for prokaryotes and get lots of ATP out of it. So that's an overview. Let's start with looking at glycolysis. And if you've had biochemistry, you might chuckle at this diagram because this is like the most simplified picture of glycolysis that I have ever seen. So you may be happy about that, maybe not. First of all, what do we call this? I have often just called this glycolysis, but because, especially in bacteria, there is an alternative glycolytic pathway where also sugar is split? If we're going to be really correct, yes, it is a glycolytic pathway, but we're going to correctly call it the Embden-Meyerhof pathway. And that's listed right there. I'm just going to write it a little bit bigger here. If I can spell it. Oh, I always forget that H in there. The Embden-Meyerhof-Parnas pathway. Okay, so yes, it is a glycolytic pathway, and yeah, we usually refer to this as glycolysis because this is very widespread. Who's doing it? Almost all organisms. Everybody's doing it. And when we name it Embden-Meyerhof-Parnas, that just gives, you know, the nod to the people that elucidated it. You can see we're starting with 6-carbon glucose. There's a lysis step. We have to input some ATP. I should mention that. There's a splitting step there. These actually aren't symmetric compounds. I'm going to talk about that in a minute. But then we end up here with some two NADHs, some ATP, and 2 3-carbon pyruvates or pyruvic acid, same thing. Okay, so let's see. Who's doing it? Where are these reactions taking place? Cytoplasm. And that's prokaryotes and eukaryotes. Doesn't matter, it's going on in the cytoplasm. Okay, so what are we gaining from Embden-Meyerhof-Parnas pathway? We're putting in 2 ATP, let's say net gains, but we're gaining 4. So we're net gains of 2 ATP, 2 NADHs. Again, that's carrying 2 electrons. Those are going to get shuttled to an electron transport system, and 2 pyruvates. So I said almost all organisms are doing this. Who's not? There are some bacteria, and here I'm going to show you a little bit more detailed picture of glycolysis here on the right. Who's doing it? Who's not doing it? There are some bacteria that actually are deficient in, I think it's this enzyme right here. It's phosphofructokinase. Maybe some of you have that memorized, but I don't. Okay, so there are some bacteria that can't make this enzyme. So essentially, you know, maybe glycolysis would start, and then, you know, this molecule is going to build up. So it's these reactions are reversible. It can go back and find the enzymes for an alternative pathway called Entner-Dodorov. So kind of confusing, kind of sounds like Embden-Meyerhof. Again, 2 different people that elucidated this pathway. This is only some bacteria are going to do the Entner-Dodorov. And again, they're only doing it usually because they're missing that enzyme. That's not strictly true. I gave an example of some organisms down here. Don't memorize those. That's not a complete list. We know that E. coli will do the Entner-Dodorov pathway under certain conditions if we grow it on a certain media. But it appears that other organisms don't have any choice, and they have to do that just because they're missing this enzyme. So if you look at Entner-Dodorov, first of all, before we look at that too closely, I wanted to point off here, right here, the lysis step of glycolysis. Again, don't memorize these intermediates, but this fructose 1,6-diphosphate, that is not a symmetric molecule. So you can't just split it down the center and get 2 things that are the same. So you end up with this DHAP dihydroxyacetone phosphate that actually usually gets converted into glyceraldehyde 3-phosphate. So that's why that's kind of glossed over sometimes. But as we'll see when we talk about, hey, what happens when lipids are metabolized, they actually make dihydroxyacetone phosphate, DHAP, and it gets shunted into glycolysis at this step. Okay, so we will revisit that later. But if you compare the Entner-Doudoroff pathway to the Embden-Meyerhof pathway, you see some similarities. But right to begin with here, there's a difference. Here we've got NADP being used as an electron carrier instead of NADH, which is, of course, not till down a little bit lower in the pathway. So remember, these electrons are going to go to power some biosynthetic pathway. They're not going to be converted into energy. And then right away here, we get a pyruvate. So we kind of are missing out on ATP and the reducing power that we normally get down here at the bottom of glycolysis for one of those pyruvates. But the bottom half here of the Entner-Doudoroff is the same as the bottom half of glycolysis. So here we're getting 2 ATPs and we're getting an NADH. But we still end up with 1,2 pyruvates. Okay, so net gains of the Entner-Doudoroff, we're getting 1 NADPH, 1 NADH. Only we put in 1 ATP and we get out 2. So we're only getting 1 ATP. So that's half the number that we get. I wrote this again for glycolysis. Is that a problem? Usually no, because usually organisms that use this pathway are doing some kind of respiration, and they're going to get most of their ATP later on in the electron transfer system anyway. So to only get 1 instead of 2 from glycolysis is not a big deal. If they were not doing respiration and all their ATP is just from this pathway, then it may be a problem, but that's usually not the case. Okay, and still get 2 pyruvates, just like the Embden-Meyerhof. Okay, so again, only certain, we actually think that Entner-Doudoroff may have been the more primitive pathway, and eventually, so organisms lost their ability to do that, maybe? Question. So right down here? Yeah, so 1 is right here. 1 comes off early, yes. So no, that's good. Because sometimes, like, see, this just has a little 2 there. And so you kind of they abbreviate instead of showing both ways, yeah. So up here at the front, where does that initial ATP come from? Just from, maybe it's got some stored up in the cell, or from, well, not mitochondria, not in bacteria. Yes. So, right, this is, hey, maybe it just metabolized some glucose and it's got 1 over here. Maybe it goes right back in and keeps it going. So, good, yeah. Yeah, so how in depth do you need to know this? I would say know what we start out with, know what we finish with, know what we get out of it. What are those net gains? Yeah, so none of the intermediates. Okay, there is another alternative to glycolysis that we'll finish with today, and that's called the pentose phosphate pathway. This is both prokaryotes and eukaryotes do this. It does go by another name, hexose monophosphate shunt. I never use that name. I always call it pentose phosphate pathway, but you might see that somewhere. When would organisms do the pentose phosphate pathway instead of Embden-Meyerhof? It's used when an organism's needs are biosynthetic. Okay, it's got lots of energy. It just needs to make maybe cell molecules, some kind of cell structures. Okay, so if we look at this again, we're starting with glucose. Right away here, again, that other electron carrier, NADP. So we're gaining 2 NADPHs. Oops. NADPHs. So remember, those electrons help power biosynthetic reactions. And then other possible gains. This is a complicated looking pathway, but it's not going to follow every arrow. It's either going to go one way or the other, and it all depends on what the cell needs. If it needs to synthesize nucleotides, then we might lose a carbon right here by a decarboxylation and end up with this 5-carbon ribose 5-phosphate. Okay, pentose sugars like deoxyribose, ribose in DNA and RNA. Again, depending what the cell needs, there might be some reconfigurations and we may end up with erythrose 4-phosphate. And that's used for cells to build aromatic amino acids. Okay. Now, humans can't make the ringed amino acids, but a lot of bacteria can. So that's something that only bacteria do. And you know, if you shuffle down through here, you've got some carbons left over, and some of those leftover carbons, like maybe glyceraldehyde 3-phosphate, are going to get shunted into glycolysis, and then maybe a little bit of energy getting out of kind of whatever's left over. Okay, so no ATP directly from this pathway, pentose phosphate. Okay, again, it could shuttle some of kind of the leftover bits into glycolysis and end up getting maybe an ATP or 2 out of it, but not directly from this pathway. We're looking for precursors to nucleic acids like the ribose 5-phosphate or the erythrose 4-phosphate in making amino acids. Okay, so a cell can be doing pentose phosphate at the same time as it's doing the Embden-Meyerhof, getting energy, and it's getting precursors for biosynthesis. Okay. So it doesn't have to be doing one or the other. That seems like a good place to stop to me. So we'll talk about what happens to those pyruvates next, how they get shuttled into the Krebs cycle, and onto the rest of respiration next time. So have a good day.